ఆన్ చేసుకోలేదు.
Good morning, everyone. My name is Monica. I'm a research scholar under the supervision of Professor G. Shamita Madam, BOS Department of Geology, Kakatiya University. Today, I would like to give my presentation on exploiting indigenous gut probiotics of Tassar silkworm anthiramycota for nutritional fortification, a comparative study with spirulina on larval growth and post-cocoon traits. In this study, we have done with the supplementation of probiotics and spirulina to the Tassar silkworm to improve the larval growth and post-cocoon parameters. Introduction. Anthiramycota is an economically important Tassar silkworm which is widely used for the silk production. Silk production depends, the quality and the quantity of silk production depends upon the nutritional value of the Tassar silkworm. Okay, and the Tassar silkworm which feeds on the Ternalia arjuna plants, which serves as primary food plants to the Tassar silkworm. Okay, the Ternalia arjuna provides the nutritional supplements to the Tassar silkworm. Okay, although Ternalia arjuna provides nutritional supplements, why we are giving probiotics and spirulina as supplements to the Tassar silkworm? Why means to improve the growth and the post-cocoon parameters. And one more thing is, not all the silkworms have the same immunity, and not all the silkworms can acquire the immunity itself. So that we are providing the immunity indirectly by giving or by providing probiotics and spirulina By spirulina, we can provide the nutrients, minerals, vitamins to the Tassar silkworm. Okay, objectives. First objective is to rear Anthiramycota on Ternalia arjuna plants, and the second one is isolation and identification of gut bacteria from Anthiramycota. I have told you that probiotics, right? Probiotics are nothing but microorganisms. From where we have isolate the microorganisms means we have isolate those probiotics, means those bacteria from this Tassar silkworm only itself. Okay, and the third objective is to evaluate the probiotic potential of isolated gut bacteria. And the fourth one is to prepare and apply probiotic supplements and spirulina solution at three different concentrations. Okay, and the fifth one is to study the effect of these treatments on lab on Tassar silkworm. And the sixth objective is to compare the effectiveness of probiotics and as well as spirulina. The fifth is itself turn into the cocoon. You can see it here, and we have done with the isolation and identification of bacteria from the Tassar silkworm. And after identification, we have determined is it probiotic or not. For the determination, we have done with the or not. For the determination, we have done with the acid tolerance assay. Is it to survive at acid conditions? Which are is it showing hemolysis or not? And the preparation of bacterial suspensions like probiotics and spirulina at three different concentrations, 1:10, 1:25, and 1:50. We have divided the worms into three different concentrations, as I said, 1:10, 1:25, and 1:50. This is the fortification, the foliar spray, and economical parameters were done with the standard protocols. And results after the we have identified the bacteria as Mammilococcus curie. After the identification, we have done with the acid tolerance assay. Our bacteria can survive at pH 4 conditions, and 80% of bacteria can survive at pH 4, and there is no hemolysis, there is no breakdown of blood cells. So it is concluded as it is probiotics. Then after we have Then after we have supplied, gave the supplements of probiotics and spirulina, and in results you can see here larval weight and larval length in at 1:25 concentration only gives the highest parameters. At lowest, yes. You can in the present study, the probiotics and spirulina both gives more parameters than the control. But comparatively, probiotic and spirulina, probiotics only gives the more parameters of larval and post-cocoon parameters. Thank you.
Session is open for questions.
Sorry.
One question since the organizers have allotted only one question. So if there are no questions, then we can proceed to the next part. Next, Arun Kumar.
Very warm good afternoon to respected dignitaries, faculty members, my dear friends. Myself, I am Arun. I am from Microbiology Department, MSc student, Bharati University. My topic is come to comparative analysis of chlorophyll, pigments, and total protein content in healthy and virus infected foods. The next introduction regarding my topic is the crop plants are the foundation for the food security. Yes, they are also foundation for the agriculture, sustainable agriculture. But the plant diseases are affecting the crop plants, and nearly 20 to 40% of crop plants are being affected in the worldwide. And that there is a graph that shows that the decrease of plant diseases, and with that many crop losses are being happening. And my study objectives are to estimate the chlorophyll pigments in infected and healthy leaves, and also to determine the total protein content, and also to compare the biochemical changes in healthy and infected plants, and also to understand the effect of viral infections on plant metabolism and physiology. And next, the sample collection I had done in the from Karimnagar Agriculture Research Center, where the plants are tomato plant, chili plant, and bottle gourd, visit gourd. These leaves I had been extracted, healthy leaves and viral infected leaves. And next, this was the experiment I had done in the laboratory with the I had first of all choose the 1 grams of healthy and infected leaves separately. After that, they were grinded with the 80% of acetone, and next they were centrifuged at the 3000 rpm in the centrifuge. And next, that centrifuge has been done for the 5 minutes. Next, the filtrate, the supernatant was collected in the clean test tubes, and for that remaining pellet were again the acetone was added, and again it was centrifuged for 5 minutes at 3000 rpm. And next, the both supernatant, the first supernatant and the second supernatant was both was both was added, and for that the 9 ml of acetone was added, and that was the readings was taken under the spectrophotometer at the OD of 660 nanometers and 645 nanometers. And for that, I had done for the chlorophyll A and chlorophyll B with the formulas. And next, protein estimation is also done with the healthy and infected use. First of all, I had weighed the 0.5 grams of healthy and infected use separately. After that, I had blended them with water, and after that, they had centrifuged it for 15 minutes at 8,000 RPM. And next, the alkaline reagent was added to the solution. The alkaline reagent was prepared with the 2% of sodium carbonate and 1% of sodium potassium tartarate and 0.5% of copper sulfate. And all these were mixed and they were added to my protein solution. So after that, they were incubated for 10 minutes. After that, I had added FC reagent. Then the color has been developed, the blue color intensity. The intensity was read under the spectrophotometer at the OD of 660 nanometers. And next, these are the results I had been observed in the protein estimation and chlorophyll estimation. These red color ones are all the infected and green color ones are the healthy plants. And here, by this, I had observed that the decrease of chlorophyll content in infected plants and decrease of protein content in infected plants. And in some proteins, the chlorophyll in some plants the protein content is not been decreased because of the stress proteins has been released. And next, this is the graphical representation of my result. And here the blue color one is the healthy plants and the violet color one shows the infected plants. And next, by this I conclude that the viral infection is running away, and because of that the chlorophyll and protein content has been decreasing in the plants. With that, the plant metabolism and physiological properties has been disturbing, so that the plant unable to give a good yield. And protecting plants means protecting life. And thank you for giving this wonderful opportunity, Dr. Sarita. Any questions?
Questions?
Any questions? Or any inquisitiveness, at least you don't ask subject questions, at least be inquisitive. You did your work out of your interest, huh?
Yes.
In future, you want to join as a scholar?
Yes, ma'am, I had it in PhD interest also.
You are basic.
Yes, ma'am. Yes, ma'am.
Currently, you are in.
Yes, ma'am.
What was good for you?
Yeah.
What did you time?
Finally, I want to prove that we need to go for resistant plants, and further we need to study on plants too. Normally, the plants I had chooses that the tomato plant, chili plant. In every day, we need the tomato and chili, particularly in our plant. So when we get hybrid resistant plants for these viral infections,
How will you proceed? How will you proceed for your PhD?
from a PhD. In the further, I will go for plant viral diseases, infections to more number of plants studying and discovering new hybrid varieties.
Now you would have put for hybrid variety also, and that would have been very great. Huh? Good. Next participant, Nandini.
A very good afternoon to all of you. Myself, Dr. Renuka Gattu. I am faculty of Vagdevi Degree and PG College. My PhD and postdoc under the supervision of Professor J. Shavita Ma'am, VOS Chairperson, Department of Zoology. So today we are going to talk about our research topic, that is nothing but molecular characterization and genomic innovations in non-mulberry silkworms. So now coming to the, first of all, we are introduced about the sericulture. So the sericulture is the silk producing agro-based industry. It is the combination of the agriculture, animal husbandry, cottage industry and textile industry. So India is the
You go for objectives of the
Okay.
study. Directly you go for objectives, results, and conclusions.
So first of all, now in our research, we are taking the 7 different type of echoraces of Tassosilicum antheria mylitta. So this is the collection of Tassosilicum antheria mylitta. So which we are taken from the 7 different states in our country. After the collection, this is the morphological and ecological studies. This is the field activities pictures, and then this is the different 7 echoraces of tussar, camoons, and marks echoraces, diversity of the echoraces. So this is the field activities, and this is our research pictures. So now coming to the material methods. First of all, we are taking the collection. So 7 different type of echoraces of Tassosilicum antheria mylitta. Then we are entering into the isolation. So genomic DNA isolation of 7 different type of echoraces we are done, especially which is the method, that is the phenol chloroform isoamyl alcohol method using the Nagaraju 1955. Then after the isolation, we are entering into the quantification. So the quantification which we are utilized in the nanodrop method. So the nanodrop is accurate model. So especially all the echoraces we are isolated. The exact quantification, the range in between the 1.7 to the 1.7 to 1.9 is the best quantification results. This is the 260 by 280 ratio of the nanodrop result. This is our isolating DNA. So this is the genomic DNA isolation picture of 7 different echoraces of Tassosilicum antheria mylitta. So in 7 different type of echoraces, each echorace we have taken 16 individuals of each echorace we are taken. So after the isolation, then go for the primer designing. So in our study, not only one type of primer. So we are taking 3 different type of primers, that is the SSR, ISSR, and EST SSRs. So in SSRs, 12 primers we are designed. Out of the 12, 10 primer combinations have shown highest polymorphism. Then the ISSR also, we are taking the 14 different type of ISSR primers which we are used. Then among the 14, the 3 ISSR primers have shown most effective and more polymorphism. Then EST SSRs also we are designed. The 24 EST primers we are designed. Out of 25 EST SSR primers, 1 primer shows highest polymorphism and single nucleotide polymorphism. Out of among the 24, 1 EST SSR primers, we identified the 60 single nucleotide polymorphisms. So this is the PCR profile. This is the pictures of our results of the SSR and ISSR. So then after the PCR, then we are entering into the phylogenetic tree. So the phylogenetic tree, among the 7 echoraces, the androlocal, we are conserve the androlocal. So the androlocal echorace, which is closely related to the GABA-BV. So we are studied the SSR, ISSR, EST SSRs. Finally, we got the results. The androlocal have genetically close to the GABA-BV. Then we are entering into the hybridization also have done in our laboratory. So this is the ISSR pictures. Among the 12 ISSR primers, UBC 861, UBC 864, UBC 807 have shown much polymorphism. So this is also phylogenetic analysis which we are using in the Binboot software. In SSR, we are using in the PopGene software. Then EST SSR, this is the picture shows the picture shows the PCR. Our target size is the 500. So after the cloning, sequencing, this is the flowchart of the EST SSR primers. So this is the picture of the first and second slide shows the double digestion. After the double digestion, then the sample sent for the sequencing. After the sequencing, we are identified. So the sequence, the sequence run in BLAST has shown the gene belonging to the 30 kD family lipoprotein. So we are identified the lipoprotein superfamily. So the lipoprotein is the more effective. The silkworm is it is present in the fat body, limb, and this is the very useful for the growth and development of the silkworm. So this is the single nucleotide polymorphism. They have identified the 60 single nucleotide SNPs. So finally we have conclude the, so from the above studies it is clear that the population evaluated not only differ the phenotypic traits, but also there is a genetic makeup also we are identified. So we are used the SSR, ISSR, ESTs, hybridization, NGS studies also done in our laboratory. So mainly we are focused on the Telangana local or Andhra local conservation of the echo rays of the Tassasilva Maltheria mylitta. So it would help in the future breeding program. Already we are developed in the F1, F2 generations also. Thank you. Thank you.
Time for question. Any?
If you have any time, please ask any questions. Thank you. Thank you, sir.
Next participant, Madhav Mola is there. B. Josna. Bhavani. Joseph. Bhavani, you can start.
Is spread into that any contaminated fruits or bites and means that is biting in saliva is the contaminated fruits are eating the human beings is that directly that virus is entered into the human body that virus is that prolonged incubation is 1 to 14 days. The 6 major proteins are there: the fusion glycoprotein, nucleoprotein, phosphoprotein. These are the 6 major proteins are there in Nipah virus. I target the G protein is eradicated totally in that Nipah virus. So that mortality rate is 40 to 70 in this Nipah virus. Enter is a movement body. This target in the that lungs. After this scheduled to the human brain. That is part. This regional disease Nipah virus is seen 2025 to 2026 with present in West Bengal. My object is my object is 4 types. I first that target the protein presentation. Which how to target the protein. That is my first object. And second one is auto blocking preparation of removable proteins. The fourth object is molecular docking parameters. In my result is I can represent gravity. Okay. Wait. This finally my conclusion is we that Nifah virus is not threatened to the human body. That is my conclusion.
Time for questions. Okay, next participant.
So now we are going to have a small break for lunch. Big break, it's not big break, only small break. For lunch. And we are here back by 2 o'clock. 2 o'clock in the same seminar hall, oral presentations are going to continue. Simultaneous poster presentations are going to